Okay, so this is the Eppendorf Centrifuge Clinic, or it's a cut-down version of the Eppendorf Centrifuge Clinic. This is something that we can offer as free training to lab managers and research groups across the UK, but we are spreading it over the whole of Europe as well. So if anyone is interested in that, then they should talk to their relevant territory manager, who would be the person that would do the actual talk. So we're going to talk about safety, we're going to talk about the forces involved, and then we're going to talk about how to take care of your centrifuge. Okay? So, centrifuges are in a lot more places than I think people realise. For example, most people don't think of their washing machine as a centrifuge, but a centrifuge is any piece of equipment that puts an object in rotation around a fixed axis and spins it in a circle. Which applies a potentially strong perpendicular force to the axis of spin. So that is why you feel the pressure when you're spinning around on a roller coaster, the same as the little roundabouts on the playground, things like that. But what that does is build up forces really quickly, and we'll talk about later about how much force that actually is, but it's a huge amount of force. So you have to make sure you're looking after them properly, because if you don't, you get this. So this is not an Eppendorf centrifuge. This was a floor standing centrifuge, and we don't make those, but this is what happens when you crash them, and it's not nice. There was a case once at a university in the UK about 10-15 years ago where they had several floor standing centrifuges in a row. Someone made a mistake with one of the ones on the end. The rotor left the centrifuge, took out the two centrifuges next to it, went through a wall and embedded itself in the wall on the other side of the corridor. So, a lot of force is involved in this, and you have to be really careful to make sure your centrifuge is being used properly so that it stays safe. So the first thing that we'll talk about is space. So, all centrifuges in the European Union have to conform to this particular regulation. This means that we have to actively crash our centrifuges, and we have to make our guarantee that our rotor chamber is crash resistant. So what that means is when it crashes, the rotor has to be kept within the external box of the centrifuge, and the external box of the centrifuge can't move more than 30 centimeters. This is why in all the manuals we say, please leave 30 centimeters around your centrifuge, which is promptly ignored by everyone in any lab ever. So what we at least try to say is please don't put anything expensive within 30 centimeters of your centrifuge, because then if you do crash it, at least you're not taking anything pricey out with it. Other safety features that most centrifuges nowadays include are things like automatic rotor recognition. So different rotors will have different restrictions in terms of the speeds and the volumes that they can hold, and the centrifuge will know what those are and automatically put restrictions on it so that you can't spin it too fast, for example. And most of them nowadays also have automatic imbalance recognition shutdown. So the centrifuge will start to spin up. If it's not balanced, it shuts itself off before any forces can build, or two larger forces can build up, and it gets unsafe. So, always load your rotors symmetrically and balance the weights. Hopefully, it's nice and obvious why this is bad and this is good. And the same for these ones. Can anyone spot what's wrong with that one? That's right, you have to answer questions. Yeah, there's no bucket. And you can all see that obviously, although these three are nice and even, this one really isn't, and that's why you want to change it. But what most people don't realise is that the balance point for a swing-out rotor is in the middle of the bucket. They think it's just the cross arms of the rotor. So, this looks really well balanced, and that's great. But when the buckets swing out horizontally, all of the weight is going to be in the top of the bucket, which means it's not going to swing out properly, and it's not going to be balanced properly, and that's going to apply torsional forces to the cross section of your rotor, and over time it's going to damage your rotor. So your centrifuge would probably not pick up on that, it probably wouldn't complain, but it will knacker it over a long period of time. I mean, obviously this one here That's on the wrong side. If it was on the wrong side, it would be better, because then it would be like this one. But even so, we would recommend something more like this instead, because the weight of the samples is more balanced around the whole bucket, not just against the cross arms. Yes. On some rotors, you can do mixed loading of plate buckets and tube buckets, but it is always worth checking with the manufacturer before you do that. It should say in the brochure or the manual somewhere whether or not it's safe to do that with that rotor. Again, automatic rotor recognition and imbalance detection should stop you from doing it if you can't, but to be on the safe side, it's always better to check. So, never spin with empty bucket slots, even if it looks balanced. Because again, that will give you torsional twist on your centrifuge rotor, on the cross arms of the rotor, and it will damage it over time. Always make sure that they can swing up fully horizontal. That was the problem on the previous slide. Yes, they could go fully horizontal, but they probably wouldn't because of all the weight being at the top of the bucket. When it comes to things like the smaller tubes, we recommend consistency, at least, in how you put your tubes in the rotor, particularly if you're working with something like DNA or RNA where you might not be able to visually see the pellet, because then you know that if you spun it like this, you know where the pellet's going to be. It's going to be on that side of the tube at the bottom under the hinge, which means you can prepare out any supernatant without disturbing your pellet. So, but when it comes to swing out rotors in particular, one of the key things that manufacturers look at more and more is being able to adapt to variation and the vast number of different formats that scientists are increasingly working in. So, Eppendorf works with a modular system. Some of the other competitors do the same, but this is definitely what we have. This means you can support tubes of different lengths. Now, a key one for this is actually, this is quite a good example, is 5mm tubes are the same shape as the bottom of a 15mm conical. So, when you've got the top two sections taken off like this one, that's the right height for a 5mm tube, so it's fine. But if you then put a 15mm straight in it, not enough of the tube is supported on the sides. We recommend that at least 2/3 of the tube height is supported. So clearly that's not the case there. We also recommend that you are choosing the right shape at the bottom of your adapter. So a lot of adapters will come with rubber funnels or rubber mats in a different shape depending on the different tubes that are involved. This is to avoid things like this. So it's not the most obvious, but this is a centrifuge tube that's nice and fine. This one has been used without the supporting insert, and you can see it started to bow. Low speeds, this is not going to be the end of the world. High speeds, you're going to lose your sample because the tubes will break, because they're not designed to withstand those forces unsupported. This is particularly important if you're spinning glass tubes. Use the supports with the glass tubes because glass is not designed for it. So, swing out buckets are actually made in pairs so that they are perfectly matched to each other. Most people think all buckets are all the same, but they are definitely made in pairs. And so when you get a new centrifuge, try and keep them together, which almost never happens. And also, the maximum load for a swing out rotor is printed on it. It's also on the fixed angle rotors. It's in the middle. It will tell you that includes the weight of the rotor and the buckets and any adapters on top of your samples. Okay? So, one of the other things that people really need to think about going forward in terms of their use of centrifuges is aerosols. So bioaerosols is a suspension of airborne particles that contain living organisms or parts of or were released by living organisms. So this can be anything from bacteria and viruses, fungal spores, enzymes, allergens, all sorts of things. And they can be in the air for hours and hours. And that means we're all breathing it in, which is just lovely. So this is where you start to get infections for your scientists, but also it can cause cross-contamination between samples. So all these different techniques can create aerosols: pipetting and mixing, opening cultures, centrifuging and not having aerosol tight caps on your tubes breaking, shaking and shaking incubators, And then once you have them in the air, you can breathe them in, you can touch them, and you can ingest them. So obviously none of this is particularly ideal. There are the three most common lab infections that people get as contaminations from their samples are salmonellosis, brucellosis, and shigellosis. None of these are nice infections. You do not want them. So it's really important to protect your lab by thinking about what you're using your sample in, by ensuring your centrifuge rotors are aerosol tight. So on Eppendorf ones, they will say, but they will also have either a red knob in the middle or there will be caps. It is something to think about, and your rep should be asking you if you're purchasing whether or not you need aerosol tight. And also actively cleaning them when someone spills, which if any of you guys are lab managers knows never happens because no one ever owns up to it. And we also recommend that if you are spinning something where you know aerosols are going to be given off, leave it for 30 minutes for them to settle before you open them. So, we'll talk more about the maintenance and cleaning up the centrifuges in a little bit, but next we're going to talk about the forces involved. So, this is our most popular model of benchtop centrifuge. It has a maximum capacity of 3 litres. The rotor, the S4104, can spin up to just over 3,000 G. This one takes 750 mils. So, doing the maths, that means that once it's spinning, the relative weight is basically the same as a car. This is why earlier we were talking about how important it is for centrifuges to be used correctly and for them to have all the safety precautions that mean that if there was a crash, it's contained, because you don't want these sort of forces being let loose in the lab. And that's why, in the example I mentioned earlier, that rotor took out 2 other centrifuges and a wall. So, you're going to actually have to answer a question here. Of these 2, they have the same top speed and they take the same size tubes. Which one is going to be applying the most force to the tubes? Anyone want to take a guess? You're saying the 5427? So the calculation you have to do is to calculate for relative centrifugal force, which, as you can see, takes into account the rotations per minute, the speed, and also the radius of the rotor. So then when you do the actual calculations, the mini spin applies 13,000 G onto the sample, whereas at the same speed, the 5427R is applying 22,000 G to the sample. Now, we always say for reproducible results, use centrifugal force. This is also relevant if you're purchasing a new centrifuge. I am regularly asked for a centrifuge that can spin at 3,000 RPM, which is great. What were you spinning before? Because 3,000 RPM in one centrifuge is very different in a different centrifuge. And most people don't know, which means I have to work it out for them. Which is fine, but it means that your results are not going to be reproducible as soon as you swap centrifuge. So, the other things to think about with your centrifuge and looking after them, the maintenance, and making sure that you're using the most appropriate equipment for your experiments is the temperature control. So most non-refrigerated centrifuges have some sort of air cooling to stop them from overheating, but there's no active control. And the temperature in the centrifuge can be influenced by rotor load, the speed and the length of the run, and the room temperature. So the longer it's running in a warm room, the hotter it's going to get, and air cooling can only do so much. So, if you actually need temperature control, you need to use a refrigerated centrifuge. And there are 2 different ways that most centrifuges use. They use one or the other in order to be refrigerated. So, in this graph, they use pre-cooling and then continuous cooling. So what happens is they cool it down at first to the right temperature, which in this case is 4 degrees. So they cool it down, and then they keep it at 4 degrees all night. So you can see the temperature is nice and consistent at 4 degrees all night, ready for them to use at 9 o'clock in the morning. That's great. But then if you look at the energy consumption, you use a ton of energy at the beginning to cool it down, but then you're still using quite a lot of energy this whole time keeping it cold. Okay? The alternative is what we call fast temp. Other manufacturers do have their own versions, but ours is fast temp. Basically what this does is it spins up the centrifuge empty before you use it. It uses the motion of the rotor to help the compressor to cool down the rotor and the chamber faster. Okay? So this means the temperature was really warm all night because it was just at room temperature. But then it comes right down first thing in the morning, ready to be at 4 degrees at 9 o'clock. But in comparison, the energy was really, really low all night because it wasn't doing anything. And then it just peaked a little bit when you actually cooled it down. So in terms of energy usage, which is a big problem with refrigerated centrifuges, using something like fast and programmable pre-cooling is really, really worthwhile. And the other thing that most of them have now is an eco shut-off. So if it's left on for too long, it will turn itself off. So, anyone want to hazard a guess as to whether or not an aluminium rotor or a carbon fibre rotor has better temperature control? Any guesses? I'm not going to judge you if you get it wrong, so you're better off just guessing. Carbon fibre. You are all wrong! Carbon fibre is an insulator. So, for example, if you stick them in a cold room and cool them down, the carbon fibre rotor never gets as cold as the aluminium rotor, because the aluminium is metal, so it conducts the heat. One of the downsides of aluminium rotors is that they tend to be quite heavy because they're metal. So the Eppendorf ones are hollow between the boreholes to counteract this. And it's not in this presentation, but we have a great image where we've literally put a carbon fibre rotor and an aluminium rotor in a cold room for 15 minutes and then taken a picture with an infrared camera. When you look into the carbon fibre rotor, the centre of the boreholes where your sample is, or it's still room temperature, whereas the whole aluminium rotor is cooled down. Okay? So, fixed angle versus swing out rotors. There are strengths and weaknesses to both. So the strengths of a fixed angle rotor is obviously going to be there's no moving parts, which means it can go much faster, which is great because you're going to get really, really compact pellets. Unfortunately, you are going to be limited on capacity. By comparison, the strength of the swing out rotor is you can be really versatile and have huge capacity. And you can do things like plates. But there's much more stress on the rotor because of all the moving parts, which means you have to have it a lot slower and often longer. Okay. So we've seen lots of really big forces are created, and we've talked about aerosols and the stuff that your centrifuge goes through on a regular basis. Now you actually want to think about looking after it and making it last as long as possible. So, those of you who work in labs, how often does your centrifuge get cleaned? There's lots of people not making eye contact right now. That's pretty normal. We recommend you clean it at least once a week. Most people don't. But regularly clean it. You need to remove things like salt and other residues because they're just going to corrode your rotors, right? There's also particular places you should lubricate so you don't get metal grinding on metal. And you should also actively look for things like the corrosion and the cracks and the holes, because as soon as you start to get them, your centrifuge isn't safe anymore. We also recommend that you have your centrifuges serviced once a year with a preventative maintenance contract. Again, is this something many of you do? Well, there's some nods this time. That's always a good thing. It's important to get them checked because this is how we can guarantee, we ensure that they are safe to use. So, actually clean them and dry them. We say after every run, but certainly after a refrigerated one, when you're going to start to get condensation building up in the rotor, wipe the water away. Okay? Preferably with a lint-free microfiber cloth that isn't going to get into the mechanism and start causing any problems. If you do need to actively clean your rotors, you can run them underwater and fill the boreholes, but don't soak it and dry them. Don't just leave them, actually dry them, because the water, again, will just slowly corrode at your rotors. There is a selection of things that you can and cannot use to clean your rotors. Things like this plastic. It's got soft bits. It's not going to scratch anything. It's great. Please never ever use these things. You will ruin your rotors. As soon as you start to scratch them, they're not balanced anymore, and then they're knackered. So, also, please don't use dishwashers. Most cleaning agents that you're using in a dishwasher are just going to be a bit too nasty for your rotors. Not if they're going to be exposed to them regularly for extended periods of time. And as soon as you start to get corrosion and holes, they're not safe to use anymore. When it comes to our set of rotors are autoclavable at 121 degrees, and some of them up to 135 or 142 degrees. It should be in the manual that comes with the centrifuge, if that is possible. If not, ask the manufacturer and we will be able to tell you. But don't autoclave them together, because the heat, heating up the rotor and then cooling back down again can cause things to lock together, and it makes a mess and it's not easy to fix. So when it comes to lubrication, there are specific spots that you should and should not lubricate. So around the rotor chamber, so you get a nice tight seal between the chamber and the lid. Good start. On the swing out rotors, the connector points between the bucket and the rotor, always worth putting a bit of grease on them. And it's also a good idea to put some on the inside of the threads of your rotor lids for fixed angles. Don't do it on the motor cone, which is the bit on the underside of your rotor, or the motor shaft, because then they can't make proper contact, so you're not going to get proper spinning. Also, actually look after your seals. So we talked about how important it is to consider whether or not your samples need to be aerosol tight or not. They are not going to be aerosol tight if your seal is not complete. So actively look at them, actively replace them. It's not difficult. We sell them. You can change them yourselves. So these are sort of things to look out for. So most people can see the sort of scratches and some corrosion around here. This is one of the most common ones, scratches on the inside of the rotor lid and scratches on the outside of the rotor. People don't think about scratches, but actually what that's doing is making it imbalanced. Unless you can scratch it completely perfectly on exactly the opposite side of the rotor, it's not balanced anymore. We also get damage to the rotor cone and the motor shaft, where people don't put the rotors on properly, or they put them on a bit haphazardly. You get scratches, and this motor shaft is actually bent, though it's not very obvious in the picture. And we also get where people haven't greased the thread on the rotor lid, and so it's just metal grinding on metal. And all of these things are making your centrifuge not safe. So, if you are getting salt debris building up on your rotor, clean it. Check it regularly. Look for things like this and clean it up. And if you start to see daylight through your rotor, don't ever use it ever again. Ever, ever. This one is a photograph of a rotor where the customer hadn't taken it out of their centrifuge since they bought it, had no idea there was a hole in the rotor because they couldn't see light through it because they never took it out. When we went and inspected it, we could see daylight through it. So we took it off them, never gave it back. Also look at things like the debris in the buckets, because people don't look for things like that. So when we do our preventative maintenance, we offer boroscope tests. So this is basically a camera that sticks into your rotor, and we will categorize your rotor boreholes based on this system. So this is what a new one looks like. This is great. This is what a used centrifuge tends to look like. This is also fine. It's safe to use, but we will check up on it every time we do an inspection. At this point, we would tell you to buy a new one. At this point, we take it off you because it's just not safe. At this point, we would probably leave it with you, but just tell you never to use it again and buy a new one. And this one, it's coming with us because clearly you can't be trusted with your rotor. And that's it. So if you guys are interested in getting an extended version of this to give as training for any of your teams where you work, let your local territory manager know. And if you want any more information about centrifuges, there's a couple of my colleagues, Jeff and George, hanging around with iPads. They will scan you. They have centrifuge maintenance posters and some centrifuge guides. And if they run out, we have more that we can send you afterwards. Any questions? Mm-hmm. So ours aren't click lids, but we have something called click lock on the newest ones, where you only have to turn the lid through 90 degrees in order to make it aerosol tight. So it doesn't click on, but it's only a 90-degree twist. Yeah, so it's a quick lock rather than having to screw them down. So a lot of the older ones you have to screw them down, which can often lead to the seals getting damaged because people over tighten them. But the newer ones have click locks, so you can't do that. Yeah. The newer ones nowadays tend to have click lock. Well, ours are the click lock, but all the manufacturers have similar systems to make it a lot quicker. But also, if you don't need aerosol tight, Eppendorf microcentrifuges are safe to spin without the rotor lid because of the technology in the centrifuge lid. So if you were doing something like a mini prep and you weren't worried about aerosols, you don't have to put the rotor lid on to use it. So then you don't have to think. Yep, you can roll them up. You can run them at full speed without the rotor lid on them, and it's safe. It's a little bit noisier, but it is safe. It's because we have something called, oh no, my brain is now completely blanked on it. Optibol, that's what it is. We have Optibol technology in the lid of the actual centrifuge, which is one of the things that also keeps them very quiet, but it means that it's safe to spin without the rotor lid. Yeah, come to the stand and we'll show you. Anything else? Well, thank you very much. Have a good day.